Okay, this is the Professor, CBMD PhD, and today's Monday, October 9th, 2023. It is 6:45 and 31 seconds, AM Eastern Standard Time, and today's research, proteomics and cancer. So let's come to it here. New cancer drug targets uncovered using base editing and chemical proteomics. Date October 5th, 2023. Source: Scripps Research Institute. As combining precise genome engineering and protein activity profiling to methodically studying thousands of cancer genes, scientists were able to pinpoint promising cancer drug targets. The scientific investigators and microbiologists used a precise gene editing approach to alter more than 13,000 possible drug targets and see which edits affected cell growth. Integrating these data with chemical proteomic information pointed toward hundreds of possible drug targets, many of them never pursued in the past. And this approach integrates two innovative methods that together form a targeted drug discovery strategy for nominating new cancer treatments. It provides preclinical information about which of the thousands of different protein sites are most likely to impact cancer cell growth. Over the past decade, research chemists and pharmaceutical companies have become excited about a class of drug that works by permanently binding to cysteines, one of the 20 amino acids that in various combinations make up all human proteins. The unique reactive chemistry of cysteines makes them easy and ideal drug targets, but because there are hundreds of thousands of cysteines scattered among human proteins, narrowing down which ones to target with drugs is extremely difficult. Even among the few thousand proteins that have been identified as critical to cancer cell growth, there are still more than 13,000 cysteines. The scientific investigators and microbiologists needed a way to narrow down the list to cysteines that have an important functional impact on cancer-relevant proteins. The microbiologists used human genetic variation to inform drug discovery. Inspired by this approach, they wondered how they could combine precise genome engineering techniques and cutting-edge chemical proteomic tools to find the next generation of cancer therapeutics. The research team laboratory spearheaded the development of base editing, a method to precisely change DNA letters, then use base editing to create targeted amino acid changes in cancer cells by introducing a variety of cysteine targeted mutations. They hypothesized they could learn more about what cysteines were most relevant to cancer cells. The microbiologists edited 13,800 spots of more than 1,750 genes precisely linked to cancer cell survival. In each case, the edit targeted a cysteine on the corresponding protein. They then tested how well cancer cells with a mutation group. Moreover, they integrated their findings with new data on the druggability of these cysteines. They ultimately found that about 160 of the druggable cysteines, when edited, impacted cancer cell growth, suggesting that drugs binding to these cysteines could potentially work to treat cancer. Among the edits with the largest impacts was a change to the cancer dependency protein TOL1. Although TOL1 is known to play an important role in trimming the ends of the cell's RNA molecules, it had not been studied as a cancer drug target. However, the team showed that small molecules could be harnessed to target this Achilles heel of cancer cells. They have found compounds targeting TOL1 that show they can block its normal activity through a fascinating mechanism that is likely to impact cancer cell growth. More research is needed to show whether a drug targeting TOL1 could be useful in human patients, but the initial results suggest that the approach of editing cysteines was effective in predicting which cysteines could work as drug targets. The researchers now plan to follow up on other targets that their experiments revealed. They also say that the next generation of chemical genetic approaches are currently under development to study druggable cysteines in diseases other than cancer. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Nature of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.